Good afternoon again, everyone. Thanks for joining us. We're going to give it just another minute to fill the room here. We'll give it about another 30 seconds to fill the room. Well, good afternoon, everyone. Thank you once again for joining us for our weekly Wednesday webinar series. My name is Brett Spazzano. I'm the Vice President of Advancement here at the National Hemophilia Foundation. On today's webinar, we will discuss new hemophilia treatments, implications for coagulation laboratory. Please feel free to ask any questions that you may have by using the Q&A feature at the bottom of your screen. We do have NHF staff members monitoring these questions, which we will present to our speaker after the presentation. This webinar is being recorded, will be available to the community beginning on Friday, January 14th. Today, we will also be live streaming on our LinkedIn and YouTube channels. I'm joined today by our very own Dr. Len Valentino, and President and CEO here at the National Hemophilia Foundation. Thanks for joining us today, Len, and I'll now turn it over to you to get us started, sir.
Thank you, Brett, and thanks everybody for joining us. Good morning, good evening, good afternoon, wherever you are in the world. I wanted to talk a little bit about some of the new hemophilia treatments and implications for the coagulation laboratory. And of course, what I'm going to talk about really has important implications for people who live with and use these products to treat their hemophilia. So the slide on the right, or the photo on the right, is of a boy that I took care of probably about 2 decades ago. He had severe hemophilia A, has severe hemophilia A, and you can see there that he had massive chronic synovitis of his left knee with a hugely swollen knee. But what I want to point out is that he also has some of the other hallmarks of chronic joint disease, and that is significant muscle atrophy. And you can see the left thigh, his quadriceps muscle is significantly atrophied, as well as his gastroc muscles in his calf. So those are, the muscle atrophy is a clear hallmark of chronic joint disease. He also has a flexion contracture, was not able to fully extend the left knee. And it's hard to appreciate, but he also has an angular deformity of that knee as well. And this was resulting in chronic problems in his hip as well as his ankle as well. So this is what we're trying to prevent with new hemophilia treatments. Next slide.
So just give me one sec here.
So on the next slide, you'll see my disclosures. I have no financial relationship and nothing to disclose here. And the opinions and information that I'm presenting are my own and not necessarily reflective of those of the National Hemophilia Foundation or its board of directors. On the next slide is the mission of NHF. And recall that the National Hemophilia Foundation is dedicated to finding cures for inheritable blood disorders and to addressing and preventing the complications of these disorders through research, through education, and through advocacy, enabling people and families to thrive. And I think that this type of presentation today is one that's important because it really talks about the work that we're doing in terms of advocating and educating around preventing complications of these disorders. Although we have a goal of searching for cures, we still need to ensure that we're preventing complications like joint disease, like that boy that you saw previously, as well as other complications of inheritable blood disorders. So on the next slide, you'll see my agenda for today's talk. We'll talk a little bit about hemophilia essentials, new hemophilia treatments, and then we'll talk about monitoring these new products that are being used to treat hemophilia. So on the next slide, we'll begin our presentation today talking about hemophilia essentials. So this audience should know well that hemophilia is a rare inherited bleeding disorder that's caused by the deficiency of a coagulation factor. It's factor 8 in hemophilia A and factor 9 in hemophilia B. The inheritance of hemophilia is what's called X-linked, and it's X-linked recessive pattern, and it has variable expressivity in affected males and incomplete penetrance and variable expressivity in carrier females. So what does that mean? That means that the mutations in the factor 8 or factor 9 genes are carried on the X chromosome, such that an affected male will not pass the hemophilia gene on to his male offspring, but because of the inheritance will pass that affected X chromosome to all of his female offspring. And then similarly, a woman who is a carrier of the factor 8 gene, whether she has symptoms or not and is considered to have hemophilia based on those symptoms, or if she's completely asymptomatic, will pass one of her two X chromosomes onto each of her daughters. In 50% of those cases, it'll be the X chromosome that has the factor 8 or factor 9 mutation. And in the other half, it'll be the unaffected or normal X chromosome. The prevalence of hemophilia per 100,000 males is about 17 cases for all severities of hemophilia A, and almost 4 per 100,000 males for hemophilia B. Why aren't females listed here? Well, we don't have a really good estimate of the number of females who are impacted by hemophilia A or B. We know that there's about 3 cases of females having the hemophilia gene, either factor 8 or factor 9 mutation, for every male. So one would guesstimate that you probably could make an estimate that if there's 17 cases per 100,000 for hemophilia A, 3 times that number would be for hemophilia in women, or about 50 cases per 100,000 females. And similarly for hemophilia B, about 3 times as many. So instead of about 4, it would be about 12 females per 100,000. So we know the prevalence in males reasonably well, but I think, again, our data from the Athens data set, as well as the community counts registry, is critically important for not only surveillance, but for understanding the demographics of hemophilia in the United States. And registries like the World Hemophilia Registry are critically important for understanding the prevalence throughout the rest of the world. On the next slide, we'll begin to talk a little bit about the complications of hemophilia. And of course, 90% of bleeding episodes in people with severe hemophilia, which is defined as a factor 8 or factor 9 level less than 1 international unit per deciliter, or less than 1% of normal, occurs in the joints. Most frequently, the joints that are impacted are the knees, the elbows, and the ankles. So we know that 90% of severe bleeding occurs in these joints, and this results in recurrent bleeding into the same joint and results in what I showed you on that first slide, hemophilic synovitis. And that is the result of proliferation of blood vessels, the proliferation of the synovial lining cells of the joint, as well as then destruction of the cartilage and what is termed hemophilic arthropathy. And I'll show you on the right-hand side some of the examples of that. Significant impairment in joint function, like that boy on the first slide had, where his joint was not as mobile as his other joints were, and he had that flexion contracture and angular deformity. And all of this decreases health-related quality of life. So on the photos on the right-hand side, these are photos from my clinical practice. The first photo at the top is an example of hemarthrosis. This is joint bleeding. You can see the right knee of this boy is swollen You can't appreciate it from the photo, but it's tender to touch. He had limited range of motion. If he tried to move that joint, it was excruciatingly painful. So this boy has acute hemarthrosis. With treatment, this would resolve, and the swelling, the pain, and the discomfort would resolve relatively quickly after treatment with factor replacement therapy. The second photo in the middle is an example again of chronic synovitis. This is a boy with severe hemophilia B, and you can see his left knee is significantly swollen again. He has muscle atrophy of both the quadriceps muscle as well as the calf musculature. But again, this is in contrast to the first photo where that knee joint was warm and extremely tender, the second photo with the synovitis was mildly tender and only mildly warm. So the level of inflammation was very different. There wasn't acute blood in that joint, and this was confirmed by ultrasound as well. So this was chronic synovitis without chronic bleeding into that joint. The bottom photograph is of a radiograph, and this is the example of hemophilic arthropathy. And what you can see here is essentially in that right knee joint, complete obliteration of the joint space. Now, why do people have that joint space? If you look on the left side, which is the right of the photograph, you can see the separation between the femur and the tibial plateau. That separation, or that dark space between the bones, is where there's cartilage. When the cartilage is destroyed because of recurrent bleeding, it disappears and you have bone on bone, which is evident in the right knee joint. You can also see that there's an angular deformity of this joint. The two bones, the femur and the tibia, don't align normally anymore, and there's a sliding of the tibia on the femur. There's also overgrowth of the femoral condyles, the knobs, that you can feel in your own knee joints that results in this angular deformity. So this is an example of relatively severe hemophilic arthropathy. This is a chronic condition. This is not going to improve without aggressive treatment to prevent further bleeding and aggressive physiotherapy to strengthen the muscles, as well as regain balance and range of motion. But it's a situation that's not likely to result in a good outcome. And these joints are ones that are destined in most cases for joint replacement surgery. Next slide. Here we see the cell-based model of coagulation. And what we know about blood clotting is that this occurs in an orderly fashion, but importantly occurs on the surface of a number of different cells. Most importantly, the fibroblast or monocyte, and then platelets. So I'm going to dissect this down into a few phases in the next slide, in which you can see that initiation of blood clotting occurs on the surface of a fibroblast or activated monocyte. Now, this is important because the initiation stage is where factor 7 or factor 7A binds to the tissue factor receptor. Tissue factor receptors are expressed on a continuous basis on fibroblasts. Fibroblasts are normally not present in the circulating blood, and fibroblasts are sequestered or sealed away in the blood vessel by the endothelial cells. So the fibroblast is expressing tissue factor on a regular basis, but when there's an injury to the blood vessel lining cells, the fibroblast and the tissue factor expressed on that cell are now exposed to the blood, and hence exposed to factor 7 or factor 7A, in which that can bind to the tissue factor, and this results in the initiation of clotting through the generation of factor 10A on the surface of the fibroblast or activated monocyte. That combines with factor 5A, and thrombin is converted to small amounts. Prothrombin or factor 2 is converted to small amounts of thrombin. The monocytes circulate in the blood in an inactive state, and the tissue factor that's present in monocytes is again sequestered from flowing blood. And only when monocytes are activated by a number of different processes, including infection and other forms of inflammation, is the tissue factor then exposed to flowing blood. This was one of the important observations that we saw in terms of the inflammatory process that occurs with viral infections, including with SARS-CoV-2. The monocytes become activated, and tissue factor is expressed then on the surface of the monocyte, present in flowing blood, but also importantly, present in the lungs. The tissue factor also binds, you can see at the lower part of that, to factor 7A. Factor 7A binds to tissue factor and converts factor 9 to factor 9A. Factor 9A then has the ability to bind to the activated platelet, and we'll pick that up in a moment. So if we look to the right-hand side of the diagram, the amplification stage, I told you before that there's small amounts of thrombin that are generated. That's the small factor 2A that's present there. And this low amount of thrombin has some very important actions. First of all, it interacts with and releases factor 8 from von Willebrand factor. And the factor 8 von Willebrand factor complex circulates in the blood. And again, factor 8 is not able to participate in blood clotting because it's bound to von Willebrand factor. But through the actions of that small concentration of thrombin, the von Willebrand factor, factor 8 interaction is broken, and factor 8 is activated to factor 8A in the blood. Another important step in this amplification process is the conversion of factor 11 to factor 11A and the conversion of factor 5 to factor 5A. So these are all amplification steps, but probably the most important amplification step is thrombin's activation of platelets. And the platelet goes from an inactive to an activated form, which is now depicted on the bottom of this diagram in the propagation phase. Previously, I told you that factor 9A was generated on the surface of the fibroblast or monocyte through the interaction of factor 7A and tissue factor. That factor 9A is capable of translocating to the activated platelet and binding to receptors on the surface of the activated platelet, which then recruits factor 8A to the surface of the platelet, which creates what's called a tenase complex. That tenase complex consists of factor 9A, factor 8A, calcium, and phospholipids present in the activated platelet. The tenase complex converts factor 10 to factor 10A, hence the name tenase. The factor 10A on the surface of the platelet is bound again to phospholipids, and factor 5A is recruited to the surface of the activated platelet, and this is now termed the prothrombinase complex. And this consists of factor 10A, factor 5A, calcium, and again, phospholipids. And this prothrombinase complex converts large amounts of prothrombin, factor 2, to large amounts of thrombin, the factor 2A. The difference between the thrombin that's generated in the propagation phase versus that which generated in the initiation phase is on the order of magnitude about 1,000-fold greater in the propagation phase. So on the surface of that activated platelet, we generate large amounts of thrombin, which are then capable of converting fibrinogen to fibrin and making a stable clot on the surface of those platelets. So on the next slide, we see the defects in hemophilia A and hemophilia B. So the defect in hemophilia A, as you recall from my initial introduction, is the absence of factor VIII. Therefore, if you don't have factor VIII, you cannot generate factor VIIIA. So in hemophilia A, there's lack of factor VIII and the inability to generate factor VIIIA to then participate in the future steps of blood clotting, as we talked about. In hemophilia B, lacking factor IX creates a situation where you're not able to generate factor IXA, which is then not able to participate in the 10-ACE complex or in the prothrombinase complex on the surface of activated platelets. And hence, both of these defects result in a decrease in thrombin generation, such that the blood clotting mechanism is severely impaired, resulting in spontaneous bleeding in the severe forms of hemophilia. So this is why people with hemophilia bleed. The lack of factor VIII and factor IX precludes the generation of large amounts of thrombin from being generated in the plasma. You can see, though, that you can still generate small amounts of thrombin towards the top. And that small amount of thrombin is still capable of activating platelets and the primary hemostatic mechanism, but secondary hemostasis is impaired. Next slide. We're going to transition now to some of the newer hemophilia treatments. Now, I'm going to preface this by saying I'm not able to discuss every new product in detail, and I'm really only giving you a small sampling of the products that are available. So the goal of hemophilia treatment is to achieve health equity comparable to those without hemophilia. And I like this diagram by Mark Skinner and colleagues that was published in Hemophilia in 2018. And it really talks about the goals of hemophilia treatment to normalize lifestyle, to prevent, or more importantly, to treat, or more importantly, to prevent bleeding. And we do this through replacement of the deficient coagulation factor activity. And I'm very careful about saying coagulation factor activity, because there's a couple different mechanisms that we'll talk about to be able to do that in subsequent slides. One is to actually replace the deficient clotting factor, factor VIII or factor IX, through protein replacement. And another mechanism is through the use of a bispecific monoclonal antibody, which goes under the trade name of Hemlibra. That doesn't replace the protein, but it replaces the activity of factor VIII in the 10-ACE and prothrombinase complexes. And then we have other mechanisms of action to replace the deficient coagulation factor activity by balancing the coagulation and anticoagulant mechanisms that we'll talk about. We also need to minimize the complications and burdens of therapy. So as we're progressing towards health equity, we are trying to get to not only better treatments, but better ways to administer these treatments with reduced burden of therapy. So the next slide will show the major advances in therapy for hemophilia and some of the associated outcomes. So if we dial back to the early parts of the previous century, in the early 1900s, we were in the pre-replacement therapy era. And that includes the times up to the use of blood transfusions. Until there was a blood transfusion, there was really only supportive care. And hospital or clinic-based care was prevalent because there was really no opportunity to do home treatment. Recurrent and acute and chronic pain was the norm, and debilitating joint disease was highly prevalent. So most boys with hemophilia had severe joint disease and died at an early age. So the mortality was very high, from uncontrolled bleeding. And this was the situation until blood transfusions were administered, which then really provided only minimal treatment of bleeding episodes. But it did result in some important advances. And that was, we learned through the use of blood transfusions that plasma was critically important to the treatment of hemophilia, and the development of cryoprecipitate by Dr. Judith Graham-Poole was a seminal event in the treatment of hemophilia. Replacement therapy began first with on-demand treatment, the treatment of bleeding as needed when bleeding occurred. An effective bleed treatment and prevention was key during the replacement therapy era. Home-based care became possible, and then high to low intensity regimens were developed for treatment at home. The risk of inhibitor emerged, but there was reduced mortality from joint disease, and the mortality decreased for people with hemophilia. There was the development of first standard half-life factor 8 and factor 9 products, followed by the development of extended half-life factor 8 and factor 9 products. Prophylaxis was introduced in the 1990s, and the seminal publication of Dr. Manco Johnson and colleagues in 2007 demonstrated the value of prophylaxis in children. Subsequently, Dr. Manco Johnson and other colleagues demonstrated the benefits of prophylaxis in adult patients. In that case, we weren't necessarily preventing joint disease, but we were preventing the progression of joint disease in additional joints for an individual. There was also the decrease in mortality because of the introduction of prophylaxis. As Dr. Pipe points out in this a figure from his publication in 2016, we had the emergence of what's called the non-replacement therapy era. And this was when we moved from being able to provide protein replacement with plasma-derived or recombinant factor 8 or factor 9, either in the standard or extended half-life forms, to non-replacement therapies, beginning first with the development, as I said, of the bispecific monoclonal antibody Hemlibra. And now we have other drugs in development that hopefully will be able to realize the true value of non-replacement therapy using multiple different modalities. These result in effective bleed treatment and prevention. The opportunities for home-based care, again, with low-intensity regimens that are not burdensome to patients, people living with these diseases or their families. The importance of liberation from IV infusions. For example, Hemlibra is administered subcutaneously, so the need for chronic intravenous injections is no longer necessary. The inhibitor risk has essentially been eliminated with drugs like bispecific monoclonal antibody because of not administering factor 8 or factor 9. There's reduced morbidity from joint disease because of better bleed, the definitive bleed prevention strategy. Home-based care will be utilized until curative treatments are available. Now, I'm not sure that gene therapy will be considered curative, but as I've heard from many people with hemophilia, cure is defined by the individual. And it's really up to that individual to help us understand what does cure mean for them. The inhibitor risk could be eliminated as well, and no further mortality from joint disease could be the future state once we have more, gene therapy or other corrective therapies available. And of course, we hope that we'll be in the situation with no bleeding-related mortality. So with that background in mind, let's go to the next slide, which talks about the products used to treat hemophilia. And you'll see here the burden of treatment increases at the top of the slide and becomes less and less as we get to the bottom. And we also believe that the efficacy of treatment will also be greater as we move down this figure. So the burden of blood transfusions was extremely high, requiring an individual to go to a hospital, to sit there for several hours for that blood transfusion. And much like the case with people with sickle cell disease or thalassemia, there's the chronic administration of blood results in what's called alloimmunization. And it becomes more and more challenging and eventually extremely difficult to identify blood that's compatible with that individual because of the development of this immune response. So blood transfusion is not an effective treatment, and it's quite burdensome. Plasma was sort of the next innovation. It was slightly more effective, but still required a significant investment of time and effort. When I first started my hemophilia practice, I was involved in donor-directed plasma donation to make cryoprecipitate. We had several fathers who donated plasma to make cryoprecipitate that was then administered to their sons to treat their hemophilia. This was a very effective form of treatment, but again, it was very burdensome to both the parent as well as to the child. The development of plasma-derived products occurred and was, again, an innovation in hemophilia treatment. Early in the 90s, the recombinant era saw the development of recombinant factor VIII and then recombinant factor IX products, which really began the real opportunity to evolve home treatment and then self-infusion. Modified products, recombinant products, were developed to extend the half-life. Much better extension of half-life with factor IX products. But we know now that the half-life of factor VIII is modulated by the half-life of von Willebrand factor, and attempts to sort of disassociate factor VIII from von Willebrand factor are in development. So drugs like the drug Sanofi is developing BIV001 is one of those opportunities to disassociate the factor VIII from the von Willebrand factor limit on half-life extension. Then there's been the development of novel mechanisms of action. And I pointed out the Genentech product, Hemlibra, as an important novel innovation in hemophilia treatment. There's other products such as fitusiran, and then as well, small molecules that are used to inhibit tissue factor pathway inhibitor, as well as other mechanisms of action that leverage other anticoagulant pathways like the antithrombin or protein C pathway as well. Gene therapy is on the horizon. It's likely that we're going to see the first approved gene therapy for hemophilia A and hemophilia B in the not too distant future. We'll see what gene therapy brings to the hemophilia community, but really the next generation of treatment will be newer vectors delivering gene therapy, but ultimately the promise of gene editing, of eliminating the factor VIII or factor IX gene is what many people are looking towards. And of course, there's academic labs and industry labs that are researching new gene therapies, gene editing, as well as developing novel mechanisms of action to innovate in the therapeutic space for hemophilia. So on the next slide, let's talk a little bit about some of the strategies to extend the half-life of recombinant products. This is information that most of you probably already know. We can reduce the interaction with clearance receptors through the process of pegylation. And there's several FDA-approved products that are available that use this strategy. We can also rescue the factor proteins from endocytose intracellular storage degradation pathways through the use of the neonatal FC receptor. And this follows what are called FC fusion proteins that are being used to extend the half-life of factor VIII, as well as albumin fusion proteins, which basically achieve the same purpose through the fusion with albumin. We also have enhanced interaction with von Willebrand factor, and the single chain factor VIII variants do this. And then optimizing tyrosine sulfation, the decoration that's on the surface of factor VIII, can also change the half-life. But as I said previously, this is a limited strategy because of the factor VIII half-life extension would again be limited by the half-life of von Willebrand factor to which it's tied to. We can also eliminate non-human glycans. Glycans are sugar molecules that decorate the surface of all of our proteins, including factor VIII. So the use of a human cell line to express factor VIII may be an opportunity to extend the half-life, as has been proposed by some. So there's many opportunities to extend the half-life for both factor VIII that I showed you here, but others for factor IX as well. On the next slide gives a list in tabular form of some of the new modified factor VIII products. We see here products from many of our leading industry partners on their attempts to develop new modified factor VIII molecules. I put the references on this slide, so you're free to be able to look in the literature to get more information on these various molecules to educate yourself. This slide was courtesy of Dr. Rajiv Purthy at the Mayo Clinic. And I'm really grateful to Rajiv because he has a really excellent review article that I'll cite in a couple more slides. On the next slide, you can see the new and modified factor IX products, again, that are available and that were developed by many of our industry partners here. These molecules use different strategies to be able to extend the half-life. Again, you can see the fusion with albumin, the use of the FC fragment to fuse to the factor IX molecule, site-specific glycopegylation, the use of a different cell line, and of course, the generation of polymorphisms in several of the in the factor IX chain, as is the case with trinactagon alpha. So you can see that there's many different attempts to modify the half-life of these proteins. So let's turn on the next slide to some novel mechanisms of action. And I put these novel mechanisms of action into the context of the slide that I showed you previously, the cell-based model of coagulation with the initiation, the amplification, and the propagation steps. So coagulation factor mutants are mimetics. So we talked about emicizumab or Hemlibra. There's also molecules that are termed super factor V molecules or factor 10A molecules, or molecules of factor VIII that have specific amino acid changes in an effort to enhance their coagulation activity and extend their half-life. There's also natural anticoagulant knockdown or disruption. We have a pro-clotting system. That's what we've been talking about. That's the cartoon on the left-hand slide. This is how blood clots, the initiation, the amplification, and the propagation stages. But what I haven't showed you is the natural anticoagulant pathway. And that's depicted in this slide through the use of molecules like tissue factor pathway inhibitor or antithrombin, or what are called serpins, which are molecules, again, that interact with our coagulation factors to dampen their effects. And what we see is that there's molecules of activated protein C that are in development. That is an effort to knock down the protein C pathway and balance the procoagulant and anticoagulant pathways. Tissue factor inhibitors are in development as well. Some monoclonal antibody like concizumab. And then, of course, antithrombin knockdown with a molecule like fitusiran, which is an RNA silencing molecule. This is a very exciting time to be researching both from a basic science as well as a clinical science perspective for clotting factor new mechanisms of action. And similarly, people who live with hemophilia should be very excited about the prospects for new molecules that will be more effective and less burdensome to treating their hemophilia, whether it's hemophilia A or hemophilia B. So lastly, on the next slide, I want to transition now to the monitoring of new products used to treat hemophilia. Next slide. So we have a couple different mechanisms to measure clotting factor activity. And one is called the one-stage factor VIII assay. The one-stage factor VIII assay relies on a contact activator, molecules or substances like silica, ellagic acid, or kaolin, that in the presence of calcium and phospholipid trigger the intrinsic pathway of coagulation, and ultimately resulting in fibrinogen generation and a clot. Likewise, the one-stage assay does not measure the extrinsic pathway, which consists of factor 7, factor 5, factor 10, factor 2 or prothrombin, and fibrinogen. It only measures that intrinsic pathway. So when the contact activator, calcium, phospholipid are added to the patient's plasma, this pathway is initiated, the endpoint being a mechanical or optical detection of a clot forming in the sample. And again, this slide is courtesy of Dr. Perthy at the Mayo Clinic. On the next slide, I take you through some of the factors that influence the one-stage assay. So there's different, ellagic acid is a synthetic phospholipid. There are also ellagic acid-like molecules that are present, and you can see those listed. Silica-based methods are also present, and there's different forms of silica that can be used in these assays. And of course, kaolin is another activator. There can be kaolin that's derived from pig or chicken. So depending on which reagents are used in the assays, there can be differences in the readouts of each of these assays. So there's a number of variables that go into the result that comes from a one-stage assay. So we have a couple different categories of variables. There are pre-analytical variables, and the Medical and Scientific Advisory Council of the National Hemophilia Foundation has provided excellent guidance on these pre-analytical variables and how the plasma should be collected, how it should be transported and stored before assay. And then we have all of these variables that affect what are called the analytical variables of the assay, what it's actually used, the instrument that's used to perform the assay. So lots of opportunities for variability. On the next slide, you'll see a cartoon depiction of what's called a chromogenic substrate assay. And in this, it's a much more simplified approach. Calcium, phospholipid, and factor 10A is added with or without thrombin to the patient's plasma. This results in a reaction in which the factor 10 and thrombin activate factor 9A and factor 8A in the patient's plasma. If factor 8A or factor 9A are deficient, it will create an inability to convert factor 10 to factor 10A. And this will then decrease the generation of a light source or a chromogenic substrate. So this assay is more simplified and streamlined. But again, there's multiple different opportunities for variability in these from the reagents that are used, as well as the instrumentation that's used. Chromogenic substrate assays are not widely available in the United States, but this is improving, and it's getting better as we continue using these assays more commonly. They're primarily reserved for research studies and research laboratories, but we're beginning to see more clinical use of chromogenic substrate assays. On the next slide, I have a comparison of traditional and novel hemophilia therapies. And what you can see here is, when the slide changes, we'll see it. So the factor VIII versus non-factor approaches. The top row shows factor VIII differences. The second row is the emicizumab or Hemlibra, and the bottom row talks a little bit more about fitusiran and anti-tissue factor pathway inhibitors. And what we can see in this slide, the factor VIII assays are effective in hemophilia A. You would have to use a factor IX assay in hemophilia B. So knowing the patient's diagnosis is extremely important. These would work in patients without inhibitors. And it's important to recognize there will be peaks and troughs. So the timing of the sampling is incredibly important. And of course, this is an intravenous dosing regimen with factor VIII. In the second row, using emicizumab as the example, again, this would only be an effective monitoring for people with hemophilia A, as Hemlibra is not effective in those with hemophilia B. This would work in both people with and without factor VIII inhibitors. There would be no need to assess the patient in terms of peaks or troughs, because there's stable pharmacodynamics of these products. And again, this is a subcutaneous dosing regimen that decreases the burden of the treatment. For the other novel mechanisms of action that are fitusiran or anti-tissue factor pathway antibodies, these again would be effective in both hemophilia A and B. Patients with and without inhibitors could be monitored with these testing. And again, there's no need to test at specific times because there aren't peaks and troughs. And again, like the emicizumab or Hemlibra, these products are administered subcutaneously. So I think that the traditional and novel therapies, there are some significant advantages in terms of being able to monitor patients with and without inhibitors and not needing to rely on specific timing of peaks and troughs to collect this blood sample. On the next slide, I just want to briefly talk about what's called thrombin generation assay. And in the initial slides that I showed you of the cell-based model of coagulation, the goal was thrombin generation, and thrombin is capable of then converting fibrinogen to fibrin. So many people ask, why don't we just measure thrombin generation directly? Well, we can do that, and we do that through what's called a thrombin generation assay. And this is an excellent review that was published by Peter Lenting in 2020 about laboratory monitoring of hemophilia A treatments. And we can see that we can measure the impact of Hemlibra using a thrombin generation assay. We can also use thrombin generation assays to measure antithrombin knockdown using a product like fitusiran. And finally, we can also use a thrombin generation assay to monitor the effect of anti-tissue factor pathway antibodies and small molecules. The challenge with thrombin generation assays is, again, standardization and widespread availability. So this is not something that clinical laboratories use and can perform on a regular basis. So thrombin generation assays are currently restricted to use in primarily research laboratories. So on the next slide, I'll give you a few take-home messages. So there's variability between various one-stage assays, especially for modified and unmodified factor VIII concentrates that are dependent on the assay system, the reagents that are used, as well as a number of pre-analytical variables. There's variability between the one-stage and chromogenic assays. I didn't show you data on this, but it's very important that the clinicians inform the laboratory team about what products are being used so that this variability can be explored. The optimal one-stage assay reagents for monitoring long-acting factor concentrates varies with the molecular modification of the protein. So depending on what factor concentrate is prescribed, the optimal reagents may need to be modified to be able to appropriately measure the effect of that. Chromogenic reagents provide the most consistently acceptable estimates of factor levels. Again, chromogenic assays are limited in availability. Data from field studies and package inserts are critically important to inform clinicians as well as laboratory experts regarding the assays that are used. And people who use these products should be informed and should be knowledgeable about the product that they're using and the tests that should be used to monitor their product. So it's important that people living with hemophilia talk to their hemophilia treatment center doctors about what assays should be used. And of course, the bottom line is enhanced communication between the laboratory professionals, the hemophilia treatment centers, and of course, with patients. So with that, I'd like to end and offer the opportunity for questions.
Thank you so much, Len. Appreciate it. And we do have several questions that came in. We've got a few minutes, so we can get to a couple of them here. Excuse me. The first one comes in from Aaron, and thank you, Aaron. It's good to see you on here. She asks, thank you, Len, for providing these Wednesday webinars and for your dedication to research, acknowledging NHF's commitment to finding cures for inheritable bleeding disorders and to addressing preventing complications of these disorders. She asked, and this was before you got to this one slide, and she corrected it there, but she asked, how is cure defined? Complete eradication, is this possible given the germline origins of hemophilia? And then her addition to that, after you explained, was to although you have advised that a cure is defined by the individual, how does NHF define a cure as part of its mission statement? This has come up a couple of times. I know this might be a difficult question, Len.
Yeah, I think it's a question that currently doesn't have an answer. Maybe in 50 years, when gene editing advances, currently there's a moratorium on germline gene editing. So the ultimate correction of hemophilia and elimination of the gene from the germline of an individual that would then preclude passing that hemophilia gene on to offspring is decades away. There's ethical considerations. There is a number of scientific and research questions that have to be addressed. So I'm hopeful that, it may be in the next 50 years, germline gene editing for hemophilia and other inheritable diseases may be a true prospect, but we're far away from that at this point. Non-germline editing is happening. We've got a number of academic research laboratories and biopharmaceutical companies that are investigating non-germline editing as a therapeutic for many diseases, including hemophilia, as well as sickle cell disease and others. I think advances in gene editing outside of the germline is something that will come to fruition in the next maybe 10 to 15, 20 years. But again, there's a lot of research that needs to be done there. There's a lot of ethical concerns around gene editing as well that need to be addressed. But I think more importantly, we need to work with all of our subject matter experts, people who live with inheritable blood disorders, to help us define what is a cure. And a cure for one person may not be the same for the next person. So it's really having that open mind to what entails a cure for you versus another person or the other person that's sitting next to you.
Great. Thanks, Len. Got time for a couple more questions. Next one that comes in. What else do you recommend patients ask about or look at when considering these options beyond just factor levels? What else should be more consistently measured in clinical trials so patients can really compare apples to apples?
Well, I'm a strong proponent of what's called shared decision making. And I believe that this begins with a conversation with an individual who lives with this disorder, and their healthcare professional, their team, including not only their physician, but nurses, social workers, physical therapists, and others, about what the optimal treatment approach is for that individual. I'm a big proponent, again, of using the shared decision-making to individualize the treatment strategies for any one individual patient. So after the clinician and the individual and their family members reach a consensus on what is the optimal treatment, then it's about ensuring access to that treatment. And that access includes ensuring reimbursement and payment for the treatment, as well as other components of, for example, we're spending a lot of time right now thinking about gene therapy and how it will be administered to people in the community, using potentially a hub and spoke model. But again, I think it begins with a dialogue between the patient and their family and the healthcare professional team. And then once that optimal treatment decision is arrived at, there's a number of other steps that have to be taken. So it's not a simple, conversation. It's probably a process that occurs over an extended period of time.
Great. Thanks, Len. We got time for one more question, and I want to get it from
Let me just add one other thing. I think our Steps for Living program has some really excellent tools around shared decision making. And I think going to the NHF website, you can find some really good tools to help guide you in shared decision making.
Great. Thanks, Len. And we got time. We're at the top of the hour here. I think we've got time for one more question, and I wanted to grab it from the audience here. Says, I'm a female with factor 8 mild, wanted to use a long-lasting product, but understand that they are not much longer lasting. Would prophylactic, but physician is afraid of overclotting. Can long-term microbleeds cause arthritic changes?
So to the last part of that complex question, yes.
Yeah.
I'm a big believer in subclinical or microhemorrhages that go undetected by the individual. And that's one of the real benefits of prophylaxis, to eliminate those subclinical bleeding events that can lead to long-term joint damage. Regarding the specific product and regimen, I'd strongly recommend that you talk to your hemophilia treatment center staff about what's optimal for you.
Great. And thanks so much, Len. I know we're at the top of the hour. We've got to go here. So I'd like to thank you for taking the time to join us this afternoon and share this information. Appreciate your time and expertise as always, sir. And I'd like to thank each and every one of you for joining us for our Wednesday webinar series. Please note that this recorded webinar will be available on Friday, January 14th at hemophilia.org under the events tab, where you can find our other archived webinars as well as a schedule. Thank you so much again for joining us, Len, for presenting, and everyone have a wonderful day.